Yes, I grew up in Kent, near Canterbury, in a small town called Ashford. My father was an MD. He was the general surgeon for the local hospital. And it was really there that I became interested in science. I got my start in cancer research was not by, it wasn't a deliberate choice. It was just the way it worked out, because I had to come to La Jolla, and quite a transition from Cambridge to California, very much more relaxed lifestyle, and quickly adopted, the local habits. I grew my hair long and started exploring the wilderness. The extra experiment that sort of led to discovery of tyrosine phosphorylation was that we were looking to see whether this protein, the polyoma protein, added phosphate to either serine or threonine, and did a routine experiment where you cut the protein up after you've used radioactive phosphate to label a protein, cut it up into single amino acids with boiling in strong acid and separating the resultant amino acids, in our case, by putting an electric charge across a plate covered in a thin layer of cellulose. And when I did that experiment the first time, I'd been too lazy to make up a fresh buffer for this separation by electrophoresis. And it turned out that the radioactive spot from this polyoma protein, it didn't migrate with either the phosphorylated serine or phosphorylated threonine. And so it was an accident, if you like. I wasn't looking for it, but it has led, to the discovery that the human proteome has 90 tyrosine kinases, and it's a very important cellular regulatory system. It's a pretty atypical cancer center, I would say, but it's well respected. The main difference is that we don't really have a space that is the cancer center. The cancer center is distributed around the institute. It's a center without walls. And we've been very sort of eclectic about the sort of science that we think could be important in cancer research. So we've always had people working in areas with organisms that most places wouldn't consider cancer research. We can point to a whole series of discoveries at the institute that have led to either sort of new diagnostics or new treatment strategies. Not that we're developing any treatments. We're simply finding things that other people can then exploit to try and develop new therapeutic approaches. So we've already got two focuses. One is pancreatic cancer, which we began around 12 years ago now, and showed that if you neutralize LIF with an antibody that's administered to the tumor-bearing mice, that you can slow the progression of the tumor. So that sort of led to a lot of interest from others in targeting LIF as a new treatment modality for pancreatic cancer, which is really sorely lacking in any effective treatment. An antibody against LIF is in clinical trials. It's good to see that what we did in the Salk was translated into potentially a new type of cancer therapy. Histidine, like tyrosine, has a ring structure, and it was known already that it could be phosphorylated, and that's what I got my R35 NCI Outstanding Investigator Award, was to pursue these studies on histidine phosphorylation in cancer. And we've had a couple of papers showing that it might play an important role in liver cancer and in neuroblastoma.